Hi, my name is Brielle Drazabul, and today I'll be presenting PD-1 signaling and HIV latency in regards to T-cell exhaustion. HIV latency is a big obstacle in creating a permanent cure for HIV infection. So first, a little bit about HIV. It's a retrovirus, meaning that it takes its RNA genome. Oh my goodness! Its RNA genome and uses host cell machinery to reverse transcribe it into DNA, integrate the DNA into the host cell genome, then use the host cell machinery again to translate other viral proteins to create a virus and leave the cell, spread, and infect other cells, especially immune cells. That's why when you hear of those unfortunately suffering from HIV and AIDS, the low CD4 count, CD4 cells, T cells are one of the main reservoirs of HIV, or main cell types that get infected with HIV. And HIV has this ability to become silent, transcriptionally silent in cells, where the host cell isn't actively reproducing RNA virus or bio-RNA or protein, viral proteins. And because HIV has this tendency, a population of cells that are transcriptionally silent with HIV are known as HIV reservoirs, and since they don't actively release a virus, the treatments that are used like ART cannot find these HIV reservoirs, so the reservoirs persist indefinitely, essentially, which is why HIV is still a permanent issue today. And yes. Oh, right. And the red is my class activity. So, big misconception is does latent HIV contribute to viral load? Do any of you guys have? Do you think it does, or do you think it doesn't?
I was going to say no, but now I might say yes.
No, you're right, it is no. Understanding this is crucial for my presentation, but yes, only cells that actively transcribing virus contribute to the viral load. Yeah. Okay, so now we go into T-cell exhaustion and PD-1 expression. So in chronic HIV infection, T cells are stimulated or have virus silent for long periods of time, and over time, yes, the cells become transcriptionally silent, but because they haven't been able to eradicate the low inflammation that's caused by HIV infected cells, they enter a dysfunctional state known as T-cell exhaustion, where they lose the ability to proliferate, activate, and destroy cells that are actively replicating HIV. And a big receptor in T-cell exhaustion, marker, I guess, in T-cell exhaustion is a PD-1 expression, where when the T-cells are exhausted, usually there's a higher expression of PD-1. And research has shown that typically there's a unique relationship with PD-1 and HIV, as I'm going to relay here. My first image, the image on the left, image B, basically shows the amount of PD-1 that's expressed on the different T cells that are infected with different viruses, CMV, EBV, and HIV virus. And this is basically showing that the interesting relationship between PD-1 expression and HIV infected cells, or especially in HIV infected cells, there's a higher amount of PD-1 expressed comparison to the other. Graph C is basically showing that PD-1 expression, a higher amount of PD-1 expression is related to antigen exposure generally, and then on the left, it's specifically HIV has a higher PD-1 expression. But this just shows correlation. This doesn't necessarily show what PD-1 does. So to answer that, we go on to the next slide. So this shows, this elaborates more on the effects of PD-1 expression, which basically on the left side, it shows the amount of RNA copies detected in the total CD8 cells population expressed with PD1 expression in relation to PD1 expression, and on the right it shows the CD4 count in relation to PD1 expressed on the CD8 total population. And as we can see, with higher PD1 expression, there's a higher amount of RNA, viral RNA detected, which contributes to PD1 enhancing viral load. And on the right, it shows inverse relationship with PD1 expression and CD4 counts, whereas the CD4's T cell counts decrease as PD1 expression is increased in the population. So this just shows that PD1 is not just a marker, but it's directly tied to disease progression, to mechanism of disease. So based on these graphs, would high PD1 expressed in T cells be like a good target for HIV or a bad target for HIV strategies? Good, yeah, good. I mean, I wouldn't be talking about it if it wasn't, but yeah, a good strategy. I mean, CD4 is often related to HIV and AIDS, so the fact that we kind of see a correlation here with PD1 and low CD4 counts, PD1 expression, we might be able to use that to find a cure for HIV. Okay. This next figure shows the number of T cells that, oh yes, so we know that PD1, there is a correlation with PD1 and CD4 count and viral load, but now we need to know if PD1 causes this reaction or if it's just correlation, basically correlation versus causation effect. So this graph is basically showing the amount of calcium expressed or detected in relation to the varying expressions of PD1. Calcium basically is an indicator of activity, T-cell activity. So we're basically measuring TCR activity on the T-cell population. So as you can see, PD1, as it is more expressed in the T-cell population, there is a significant drop in T-cell activity. Yes, so this kind of suggests that PD1 directly weakens the TCR signaling cascade and provides a mechanistic foundation for exhaustion. So this explains the hows this explains the causation effect, but how exactly does it lower T-cell activation? And this is the next graph. This graph basically shows the dose response of calcium on the y-axis in comparison to the PMHC complexes, which is basically the peptide MHC interactions of the T cell and the APC, and the presence and absence of PD1. So this response curve goes in PD1 without PD1L. PD1L is the ligand expressed on APCs, but without PD1L expression, this shows the normal calcium influx curve. But with PD1L expression, meaning PD1 activation, the curve is slightly shifted to the right, indicating the threshold is, it shifts the activation threshold. So more antigen is required to initiate the same level of activation as without activity, PD1 activity. So this shows that normally a T cell needs a few PMHC complexes, and now it needs 3 times more. The 3 times is just indicated in the literature. So if PD1 raises the activation threshold, the activity of the T cells, T cells not as active. So what would happen to important cytokines like IL-2 and TNF alpha? What would happen to the cytokines as the T cell activation drops? Yeah, it would decrease. Yeah, they would not be able to release important cytokines to help combat HIV infection. And especially some cytokines like IL-2 and TNF alpha are dependent on strong TCR signals, so they're one of the first to drop. So PD1 now affects T cell function, but T cell function, lack of activations, there's still a gap between T cell and HIV latency. So let's bridge that gap. HIV latency requires transcription, so now we need to look within the cell, how specifically does PD1 correlate to HIV-1 chronic infection. So this next image basically shows the MAP kinase pathway being used in PD1 expression. And the graphs here basically show the quantitative effects, I guess, of the amount of proteins being detected in the varying expressions. I don't know if you guys can see them, but the varying expressions of PD1 expression. So as you can see in this first graph, which is very tiny, but as PD1 expression increases, the amount of phosphorylated EK2 basically decreases, and the same trend is noted in phosphorylated NF-kappa beta, P65, and then P38 MAPK as well. So this basically shows that with PD1 expression, it uses the MAP kinase pathway to inactivate the T cells, promoting HIV transcription. And the black bars basically show the pembrolizumab, basically the monoclonal antibody, which shows that when you inhibit the reaction, it increases the activity. NF-kappa beta can now reach the HIV promoter, translating HIV and a possible mechanism for HIV eradication. So this is my schematic, basically showing that when you have when you inhibit PD1 expression interactions, the MAP kinase pathway is able to activate, go through successfully, transcribing the transcription factor, which can bind to the HIV promoter LTR and promote virus or release of HIV virus. So the immune system can indicate the cells that are infected with HIV and then eradicate them. But then with the expression of PD1, the transcription factor is not activated, and then the T cell is exhausted, transcriptionally silent, and the immune system will not be able to detect it. HIV is still within the system. Basically, these are the main takeaways, but if I'm running low on time, yeah. And then I have my exam questions. Thank you.